Scientists have created a working model of one of the most complicated and important structures in the human body. Let's talk about it. Malaria is caused by a parasite that infects red blood cells. A potential terrifying complication of this is called cerebral malaria, where infected red blood cells get trapped inside of the brain. It causes swelling in the brain that can be fatal, and even when patients are treated with drugs that clean the parasite out from the rest of their bloodstream, those infected blood cells in the brain still stick around. This is most likely caused by dysfunction in a structure called the blood-brain barrier. It's basically what it sounds like. Essentially, your blood carries nutrients and oxygen around your body, including into your brain, and the blood-brain barrier acts as a filter that makes sure nothing bad hitches a ride into your noggin. The blood-brain barrier is very complicated as a structure. It consists of all kinds of different cell types that interact in very complex ways that we're kind of still trying to work out. Recently, a group of scientists created what appears to be a very faithful model of that complicated structure, and it's allowing them to pinpoint how exactly the malaria parasite sneaks its way into the brain. Step one was making a microvascular model. They created a physical framework of tubes through this jelly-like substance. They then lined those tubes with the kinds of cells that normally line your blood vessels. Next, the scientists added in the kinds of brain cells that are also known to help form the blood-brain barrier, forming a tiny little blood-brain barrier in a dish. This new model with the brain cells was way less leaky than previous models, which have focused more on the blood vessel side of things. And once they knew that the model was working, the scientists could use it to start figuring out how the parasite does its thing. They found that things started getting screwy when parasites actually left red blood cells in order to go find new blood cells to infect. This stage of the parasite's life cycle is called egress, and the scientists realized that this process releases chemicals that mess with blood vessel cells' ability to hang on to their neighbors. Basically, all your DNA-containing cells have the same genes, but different genes get turned on and off in different cell types depending on what their specific jobs are. Cells that make up your blood vessels have the make a protein that glues you to your neighbor gene cranked way up because you don't want your veins and arteries to leak. But the chemicals that get released during parasite egress appear to trick those cells into turning that gene way down. So those sticky proteins don't get made, they come unstuck from their neighbors, and the whole system springs a leak. So now we know more about how malaria works, which will make designing treatments for it a lot easier. But the model itself is also a huge breakthrough on its own. See, the blood-brain barrier doesn't just keep out bad guys. Sometimes it also keeps out good guys that it doesn't realize are good, like synthetic drugs that we need to get into the brain to treat all kinds of disorders. Having a ready-made, accurate model of a blood-brain barrier in a dish means that we can more easily figure out how to formulate drugs that are better at getting across it, which will save countless lives.